Hello, pharmaceutical science students, and welcome to your next lecture for the cell biology part of your BCH1118 course. I'm going to be moving on to the next topic today, which is all about how cells reproduce, the cell cycle, cell division, and then we'll look at programmed cell death. And I'm going to cover this over two lectures. So we'll be starting off with the cell cycle and cell division today, then we'll be continuing with other sorts of cell division and cell death on Monday. Okay, so this is the bit of your curriculum that we're going to be covering in these two lectures: cell life cycle, divisions, phases, regulation, and cell programmed death or apoptosis. Okay, so I hope you remember that we have already discussed cell theory. The fact that cell theory has three different parts. All organisms are composed of one or more cells. The cell is the basic unit of structure and organization in organisms, and all cells come from pre-existing cells. And this is the part we're going to be talking about today. How do cells come from pre-existing cells? How are cells able to reproduce themselves? Okay, so today I'm going to be talking about the cell cycle and mitosis in eukaryotes. So I hope that you remember that there are two basic types of cell: eukaryotes and prokaryotes, and the big difference between them is whether or not they have a nucleus. Eukaryotic cells have a membrane-bound nucleus. The genetic material, the DNA, and all the proteins that organize the DNA are inside a distinct cell structure called the nucleus. In prokaryotes, the genetic material is free in the cytoplasm. So today we're going to be talking about eukaryotic cells and how they are able to divide. Okay, so we'll look next time at how prokaryotes divide. It's a process called binary fission, and it's a simpler process than eukaryotic cell division. Okay, so I'm going to show you a video now, which I hope will help to introduce this topic to you and give you some visual ideas about exactly what we're talking about. Okay. Okay, I hope you enjoyed that video, and I hope that it's given you a sort of mental visual image of some of the things that we're going to be talking about. You may already have covered some of this material in your A-level, cell cycle, cell division, mitosis and meiosis, but I hope that today I'm going to be giving you more details and that we'll be able to understand more about exactly what's going on during those processes at a molecular and biochemical level. Okay, so I'm hoping that that video has left you asking some questions. We saw the cell cycle described and drawn out like this, but I hope that you're wondering exactly what's going on inside the cell at each of those stages. That's the first thing we're going to be looking at today. Then the signal. How is the cell cycle controlled? What is the signal that prepares a cell to divide, to reproduce? What are the controls that are taking place? How is the genetic material divided between the two daughter cells? We see here this movement of chromosomes towards two poles of a cell. How does that happen? How are chromosomes moved to the two sides of the cell? Then next time you saw in the video mentioned that the cells involved in sexual reproduction are different to those involved in asexual reproduction. So we're going to be looking at the cell division that results in cells responsible for sexual reproduction, which is meiosis. And next time also, we're going to look at how cell death happens. That video actually started with talking about cells dying. So how does that happen? How does a cell die? Okay, so I'll pause there and do go to the forum and ask any questions that you might have. I'll be very happy to answer any questions. Okay, so the first of our question, what is happening in the cell at each stage of the cell cycle? So here is a diagram similar to this one. You can see G1S, G2M drawn around the cell here. Here is a similar diagram, G1, S, G2, and M that stands for mitosis. So we're going to be looking at each of these phases. So I hope you can see from this diagram that there is a stage here called G0, which is described as cell cycle arrest. So that's the first thing we're going to be looking at, G0 phase. And you'll see from this diagram that in G0, we've sort of gone out of the cycle of the cell cycle. We've stepped outside the cycle. We've left this circular pattern. So in G0 phase, the cells are not actively dividing. Two words that can be used to describe different sorts of cell in G0 are quiescent, which means quiet, or senescent, which means old. Quiescent cells are cells that are fully differentiated. They are carrying out their function, and their function is one that is so specialized that they have lost the ability to divide. So an example of this sort of cell might be a neuron in the nervous system. It's carrying out its function, but it's no longer able to divide. Senescent cells are cells which were able to divide, but now have become old or damaged and will soon undergo cell death. So both of these types of cells can be described as being in G0 phase. One thing that it's important to remember is that being in G0 doesn't mean that the cells are doing nothing, especially if they're quiescent cells. These cells are the cells that are performing their function. So in our example of a nerve cell in the nervous system, the nerve cells are carrying signals. Okay, they're performing their function, but they're no longer able to divide. Okay, so if we think of a single-celled organism, for instance, yeast, a single-celled eukaryotic organism, yeast cells can remain in G0 phase and just quietly continue their cell processes at a low level if their environment is not good, if they are not able to continue to divide. Sorry about that interruption. So yes, yeast cells can stay in G0 phase, just quietly resting while their environment is not good, while their environment doesn't allow the cell numbers to increase. That's what cell proliferation means, the numbers increasing. So the cells can just rest there in G0. So if you imagine, for instance, a culture of yeast cells growing in a beaker in a laboratory, they grow for a while and they use up all the food sources in the liquid that they're growing in. So then there's no more food. So the cells will enter G0 phase and then rest in G0 until conditions change. So if a scientist then comes and adds some sugar to that solution, so the yeast has something to use as a food source, as a carbon source, very quickly those cells will come out of G0 phase and re-enter the cell cycle. So G0 isn't always an end point. For some cells, if they are fully differentiated or old and damaged, they will not come out of G0. But in other conditions, cells may enter G0, not be actively dividing, but if conditions around them change, they can re-enter the cell cycle and start dividing again. Okay, the next phase is G1 phase. So the G here, originally it was called G to stand for gap, because early scientists observing cells dividing through a microscope could see things happening at some stages, at S phase and at mitosis, but in between those two phases, there were gaps where the cells were not visibly doing anything that could be seen at that level of looking down a light microscope. So this phase was originally called gap phase, but I think it's a better way to think of the G standing for growth phase, because what's actually happening, we now know what's happening in the cells, is that the cell is carrying out protein synthesis at a very high level. It's making sorry, it's making all the proteins that are necessary to make two new cells, to duplicate all the cell contents. We've seen over the last couple of lectures how many different things there are in the cells, all the different cell organelles, all the enzymes in the cells, all the structural proteins that give the cells its size and shape. All those things need to be doubled to make two new cells, and that's what's happening during G1 phase. We found last time also that some cell organelles have their own DNA, mitochondria and chloroplasts. And during G1, these organelles are also increasing in number, ready to be divided between the two new cells. And these organelles actually are able to reproduce themselves by a process of binary fission, which is the same sort of process that prokaryotes, bacteria, use to divide. So that's what we'll be looking at next time. So G1, a lot of protein synthesis going on, a lot of new proteins being made, the cell contents being nearly duplicated. Perhaps after division, the two new cells will be smaller than the original cell and will need a time to grow. But certainly during G1, everything is made to give each of the new cells enough material to make up that new cell. Okay, so the next phase is S phase, and the S stands for synthesis. Synthesis, making phase. And what's being made is DNA. DNA synthesis or DNA replication is happening during S phase. You'll be learning a lot more about how DNA is replicated next semester in the molecular biology course. So I'm not going to go into details of how the DNA is replicated now, but I hope that you already know that before two new cells can be made, two copies of a genetic material have to be made. The DNA has to be made into an exact copy. So in our diagram here, this is illustrated. These are two chromosomes in the cell, a blue one and a red one, that are linear pieces of DNA. So before S phase, we see them as just a straight linear piece of DNA. After S phase, they have been copied. Each one has now got a copy. So they're illustrated as this X shape with two copies of the same DNA joined at the center. Okay. So the DNA has to be replicated during S phase. It also has to be packaged and organized so that each of the new chromosomes, each of the sister chromatids that are joined together here, is exactly the same as the original chromosome before it was copied. Okay, so that's S phase. And perhaps I should say that all the proteins involved in this packaging had to be made during G1 phase. Okay, so they were ready to package the DNA when it was replicated. G2 phase, so again, this looked like a gap where nothing seems to be happening during the cell cycle if you're just looking through a microscope. But again, there's a lot of protein synthesis going on. And during G2, the cell tends to grow to be larger because it's now got twice as much genetic material. It's made a lot of new protein. So the cell volume has to expand to fit all those new things into it. It's still carrying out more protein synthesis, and it's also carrying out metabolism at a high level. It's storing energy that it's going to be using for cell division. The other thing that's going on is that the DNA that was made during S phase is being checked to make sure that the DNA replication has happened correctly, that the new DNA is exactly the same as the original DNA. So we'll be talking about checkpoint mechanisms in a minute. So before the cell can go on to the division phase, to mitosis, the cell checks the DNA and it stops in G2 if there's anything wrong with the DNA and waits until that problem can be corrected. Okay, and then M phase or mitosis, this is the actual stage of cell division. So mitosis is where the genetic material is divided between the two new cells. So here you can see that we have the X-shaped replicated chromosomes, and here at the end of mitosis we've got two new cells, each again with a single linear chromosome. And in between this stage and this stage, there are a series of steps, which are the stages of mitosis. And then the final stage, well, it's actually after mitosis, is called cytokinesis, which is the actual dividing of one cell into two new cells. Okay, so we'll be looking at the details of each of the stages of mitosis in a few minutes. So something else you might have noticed in this diagram is that these three stages, G1, S phase, and G2, are labeled as interphase. So these three stages make up interphase, the phase in between active cell division. Okay, so during mitosis and cytokinesis, the cell can be seen to be dividing. In G1, S, and G2, it is not actively dividing, so this is called interphase, the phase in between cell division. So if we talk about an interphase cell, we're referring to a cell that could be in any of these stages. Okay, so how long does this all take? Well, for a mammalian cell in cell culture, so that's growing in artificial conditions outside a multicellular organism, the cell cycle lasts for about 24 hours. So the cells divide about once every day. And of that time, 23 of the hours are spent in interphase and 1 hour is spent in mitosis and cytokinesis. Okay. So pause and you can ask any questions on the forum, please. Okay, so how is the cell cycle controlled? How are these ordered stages set about and made to happen in the right order at the right time? Well, we saw in the video that it begins with a chemical signal to prepare for reproduction. So what is that signal? Well, for a multicellular organism, if you think of a growing fetus in the womb, for instance, the chemical signal might be a growth factor or hormone. So I'm sure that you're going to do embryology soon, so you'll be learning about the way that a series of signals are released that tell certain organs to grow and develop and to form at different times during development. So that sort of signal can tell the cells that receive and respond to that signal that it's time for them to divide, to reproduce. If we think of a single-celled organism again, if we go back to the idea of yeast in a culture, in a laboratory, we talked about adding more food source, so the signal that the cells receive might be the presence of that food source. We're now finding that the cells are able to have enough food to divide, so the signal that tells the cells to divide is the food itself, the presence of the food or something connected to the food in the environment around the cell. Okay, so how is the cell cycle controlled? Well, once a cell enters this cell cycle, it has to go through the whole of the cell cycle in the correct order. So if we think of this as something a bit like a clock with a finger going around it, this finger has to go round the whole cycle in the correct order. It can't jump from G1 to G2 without going through S phase. It can't jump from M to G2. It can't go backwards. It can't go from M to G2 to S. These stages have to happen in the right order and in the right sequence. It can't go backwards and it can't miss a stage. So at each of the stages, there are some questions that are being asked at the end of the stage. So if we start here in G1, the cell has been growing since its original mitosis that produced the cell. The cell is growing and it now reaches a stage when it's become big enough to proceed to S phase. And at this point, there is what's called the start checkpoint. So this is where the cell really makes the decision of, is it going to take part in cell division, or is it going to wait longer, or is it going to enter G0 and not divide at all? So the start checkpoint, the big question that's being asked is the environment favorable? Is the conditions around the cell good to allow the cell to divide? So if we're thinking about cell development in a multicellular organism, an a favorable environment might be an environment where a hormone signal is telling that cell that it's time to divide. If we think of a yeast cell in a cell culture, the question might be saying, is there enough food for more cells? Should the cell divide to use more food source? So start checkpoint, is the environment favorable? So here is the S phase, the synthesis phase where DNA is being synthesized. So after DNA synthesis, we enter G2 phase, and there is another checkpoint at the end of G2 before the cell enters mitosis. So the questions being asked here, again, the question is being asked is the environment favorable? Is the signal telling the cell to divide still present? Is the food source still present? And the second big question, very important question that's being asked is all the DNA replicated? Okay, so if something has gone wrong with S phase here and the DNA is not fully replicated, the cell will wait at this point until the DNA replication is finished. If there is something wrong and DNA replication can't finish, the cell will stay here in G2 and not go on to divide. Because if the DNA wasn't all replicated and the cell did go on to divide, one of the new cells wouldn't get a full set of the genetic material. And that would be bad. It would produce either a cell that would just die because it wouldn't be able to produce everything it needs to live, which would be a waste of resources for the growing cells, or even worse, it might produce a cell that is so very wrong that it would damage the other cells around us. Okay, so is all the DNA replicated? So if both those questions can be answered with a yes, the cell passes the G2M checkpoint and goes on into mitosis. So we'll look at the order of mitosis in a minute, but there is another checkpoint during mitosis. It's an important stage where the question that's being asked is, are all the chromosomes attached to the spindle? And we'll see in a little bit why that is so important. So if that question can be answered with a yes, and the chromosomes are all attached to the spindle, the cell can continue past this checkpoint from metaphase to anaphase and go on to cytokinesis, the actual cell division. Okay, so a series of checkpoints making sure that the cell is doing everything correctly. Now, please remember that what I'm teaching you about is what should happen. What usually does happen, the correct behavior of cells, that usually just happens so smoothly that we don't notice. Some of you might be thinking, but we've been taught about cancer cells. Cancer cells can divide when the DNA hasn't been completely replicated. Cancer cells can divide when the chromosomes are not attached to the spindle. You're completely right, but put those thoughts to the back of your mind for the time being, and remember that most of the time, things do happen as they should. The cell cycle does happen correctly. Okay, so here's just a summary again of the three checkpoints: G1 checkpoint or start checkpoint, G2 set checkpoint, and metaphase to anaphase or M checkpoint. And if everything isn't right, the cell cycle does not continue until the problem is resolved, is fixed. Okay, so let's have a look in more detail now about just how these checkpoints are controlled. So here's another diagram showing more or less the same thing, but with just a bit more detail on it. So here is G1 phase with cell growth happening, and here is the G1 checkpoint. Here is S phase with DNA replication happening, and the G2 phase with the G2 checkpoint, and here is mitosis with the M checkpoint before the cell can actually divide in cytokinesis. So what actually makes this happen at a molecular level is that a number of enzymes that are needed to carry out the functions to allow the cell to pass these checkpoints to go on to the next stage can exist in two different forms: a phosphorylated form and an unphosphorylated form. So now I hope that you're remembering what you've learned about amino acids and the way that amino acids can be modified. And one of those modifications is that a number of different amino acids can have phosphate groups added or removed. from particular amino acids. Okay, so these enzymes can have specific enzymes which add phosphate groups to them. So phosphorylation is the addition of a phosphate group. And the enzymes that add these phosphate groups are called kinases. This is a general name for an enzyme that adds a phosphate group to any protein molecule, a kinase. So the kinase is a class of enzymes that carry out phosphorylation reactions. Okay, so involved in the cell cycle, there are particular subset of kinase enzymes called cyclin-dependent kinases, or CDKs. And they are the enzymes that carry out phosphorylation of enzymes which allow the cell to pass the checkpoints. So, cyclin-dependent kinases. What is a cyclin? Well, cyclins are proteins whose concentrations in the cell, which are produced in different concentrations, so their levels, their concentration in the cell rises and falls cyclically at different stages of the cell cycle. Okay, and CDKs, cyclin dependent, depending on them, kinases, are only active when they're bound to a cyclin molecule. Okay, so the cyclin dependent kinases are only able to carry out their job of phosphorylating other molecules when they are bound to a cyclin molecule. Cyclin molecules go up and down in the cell during the cell cycle. So at some times there are lots of cyclin molecules, and at other times there are very few cyclin molecules. So here is a diagram of the cell cycle, just stretched out into a line rather than in a circle. And this is what we're seeing about the levels of cyclins. So here are three different CDKs being illustrated. There's actually more than three, but three are illustrated here. One is involved in the start checkpoint to allow the G1 to S phase transition. This one is also involved in allowing the cell to enter S phase, and this one is involved in allowing the cell to enter mitosis. Okay. The G1S CDK can only be active when it's bound to a G1S cyclin, and that's illustrated here as this little blue molecule. And the level, the presence of G1S cyclin is very high during G phase, G1 phase, but very low during the rest of the cell cycle. Okay, so this cyclin is only active during G1 phase. In the same way, the S cyclin here is only active when it's bound to this orange molecule, which is illustrated with a red line here for some reason. This is present in the cell right through S phase, but not during metaphase and anaphase of mitosis, or the beginning of G1 phase. Okay, so this cyclin is active during S phase and G2 phase, but not G1 phase. Here's the third cyclin, the mitosis cyclin, which is active when it's bound to the M cyclin here, illustrated in green. And this one, the presence in the cell rises during S phase, so it's at its highest concentration at the end of S phase when mitosis is about to start. Okay, so although these CDKs are there in the cell all the time, they're only actively phosphorylating other enzymes when they're bound to their correct cyclin, and the correct cyclin is only there at different stages of the cell cycle, depending which cyclin we're talking about. If you're wondering how the levels of cyclin are increased and decreased, again, you're going to learn a lot more about how protein synthesis is controlled, how gene expression is controlled next semester in molecular biology. But basically, the cyclins are produced by transcription and translation from DNA information when they're needed, and then they're removed by protein destruction when they're no longer needed. Okay, so this is just putting into words what I've already said. So which cyclin is bound to which CDK is what makes different molecules phosphorylated by the kinases. So it's only when enough other enzymes have become phosphorylated, for instance, here at the G1 during the S phase cyclin, it's the enzymes which actually carry out DNA replication, or some of the enzymes which control that, which become phosphorylated. Okay, so let's just summarize that. For the cell cycle to proceed, to move on to the next stage, conditions have to be favorable, and the previous stage of the cell cycle has to be successfully completed. If this is the case, if everything has gone well and the cell is ready to move on through the cell cycle and reproduce, the appropriate cyclin is produced. This cyclin binds to the appropriate cyclin-dependent kinase. The cyclin-dependent kinase becomes active and phosphorylates other enzymes, which then become active themselves, and they carry out the processes that are needed for the next stage of the cell cycle. If it's not the case, if the previous stage of a cell cycle has not been successfully completed, no cyclin is produced. Therefore, cyclin-dependent kinase remains inactive. It's not able to phosphorylate any of the enzymes it should phosphorylate. So the cell cycle can't continue. In the example I used, it's not able to replicate its DNA. So this is described as the cell being arrested at the checkpoint. So if the checkpoint is not passed, if the cell cycle is arrested, the cell will try to correct the problem. It will try to make it right and fix whatever is wrong. Or if it's that the conditions are not right, if there is a shortage of food, it waits at that checkpoint until conditions become better. If something is very, very wrong with the cell, for instance, if DNA synthesis, DNA replication has gone very wrong, and some chromosomes have been perhaps even replicated more than once, for instance. The cell will then go on to destroy itself. It will enter cell death, and we'll talk about that next time. Okay, so here is a diagram just summarizing in a circular form what I've been telling you about CDKs. So here's G1, S phase, G2M with the 3 different checkpoints. Here is the cyclin that's active at that point, binding to a CDK. And this just emphasizes the need for energy from ATP at some cases, and it shows the degradation, the destruction of the cyclin proteins when they are no longer needed. Okay, so that is control of the cell cycle. So do ask any questions. So the next thing we're going to think about is the actual process of cell division, the stages that are needed to divide the genetic material between the 2 daughter cells. Okay, so this is what's happening in mitosis and then going on to cytokinesis. So when a cell enters mitosis, the cell stops growing. It's the end of a G2 phase, and the cell is now concentrating on cell division. Mitosis has a series of stages, which are indicated here by these lines. And again, they're ordered stages. Each one has to happen in the right order. And this is to make sure that the genetic material is correctly divided between the 2 new daughter cells so that each new nucleus has a complete set of genetic material. So stages of mitosis to divide up the genetic material, and then cytokinesis officially is not part of mitosis. It's the next stage after mitosis, and that's where the cell actually divides into 2 new cells. Okay, so another little video which just shows you cells going through mitosis and talks about the stages.
During the mitotic phase, a cell will undergo mitosis to form 2 new nuclei, and then divide to form 2 new individual cells during cytokinesis. Mitosis is the process of dividing the duplicated DNA of a cell into 2 new nuclei. Mitosis is split into distinct stages. The first stage is prophase. The DNA condenses, organizes, and the classic chromosome structure appears. Next comes prometaphase, where microtubules attach to the chromosomes. This step is followed by metaphase, where the chromosomes align. Metaphase is followed by anaphase, where the chromosomes separate. Finally, during telophase, nuclear membranes reappear around the 2 sets of chromosomes. Mitosis is now complete. After mitosis, 2 new cells are formed by a process called cytokinesis. During the first stage of mitosis, called prophase, we first see the classic chromosome structure. This occurs through a condensation process. At the same time, protein strands called microtubules appear from the centrosomes in animals. Finally, a structure found within the nucleus, the nucleolus, disappears. Next, prometaphase begins when the nuclear membrane is broken down. At the same time, microtubule strands or spindle fibers are growing from the centrosomes. These strands attach to a protein structure called the kinetochore. One kinetochore is attached to the centromere of each sister chromatid. Next comes metaphase. During this stage, the sister chromatids align along the center of the cell so that both chromatids face toward opposite poles of the cell. Now the sister chromatids are ready to be separated. This occurs during anaphase through a shortening of the microtubules attached to the kinetochores. Additionally, the poles of the cell move farther apart, causing increased separation of sister chromatids. At the end of anaphase, the sister chromatids have moved to the two ends of the cell. Telophase is the final stage of mitosis. It is here the components of the new cells begin to appear. At this point, the spindle fibers are broken up. A new nuclear membrane surrounds the chromosomes at the end of each cell, and the chromosomes uncoil and return to an uncondensed state. Mitosis is now complete. The formation of two cells is all that remains. Following mitosis, the cell undergoes a process called cytokinesis. First, the cell is compressed by a contractile ring that divides the cell in nearly equal halves. By now, the organelles in the cell have been replicated and are now divided between the two halves of the cell. This includes mitochondria, Golgi bodies, and the rough ER. Plant cells also have chloroplasts. Once split, the two new cells are now fully in the G1 stage of interphase and ready again to begin their growth. Let's watch the process one more time. Mitosis begins with prophase. Notice the DNA condensing into chromosomes during this stage. Microtubules appear during prometaphase, and the nuclear membrane breaks down. Metaphase occurs when the chromosomes are aligned at the center of the cell. During anaphase, the chromosomes are moving apart. The telophase stage is marked by the appearance of new nuclear membranes. This is the end of mitosis. Finally, the splitting of the cell occurs during cytokinesis. The two new cells are now ready to grow and perform their specialized functions.
Okay, I hope you enjoyed that video. That is an excellent summary of what's going on during mitosis. So if you want to go back and re-watch the video, please do, or you can find it on YouTube.
During the mitotic phase, a cell will undergo
Okay, so what we saw in that cell was the process of mitosis, the process of dividing the duplicated DNA of a cell into two new nuclei. And I hope you realized that this film clip that started the video is an actual film taken through a microscope of a dividing cell. These are the chromosomes you can see moving to two different sides of the cell, ready to split into two different parts. So that's what can be actually observed through a microscope. So we saw that there are a number of different stages of cell division given the name prophase, prometaphase, metaphase, anaphase, and telophase. And then after mitosis comes cytokinesis. So let's have a look at each of those stages. So for each of the stages, I'm going to show you a clip from the video, just to remind you of what you saw, and an actual micrograph, a picture taken through a microscope using different colored fluorescent staining. So here we've got chromosomes, we've got DNA stained in red, and microtubules, the microtubules of the spindle stained in green. So this is an actual dividing cell. This is a computer animation, a drawing of what's seen. Okay, so what's happening during prophase? Well, the chromatin condenses into chromosomes. So remember that chromatin is DNA wrapped around histones and then coiled into higher order fibers. Okay. When the cell is getting ready to divide, it condenses that chromatin even more to make very condensed structures. So this sort of chromosome is not found in cells in between cell divisions, but it's not found in interphase cells because the instructions carried in the DNA wouldn't be available to be read when the chromatin is so very condensed. This is only for cell division. And the other thing to notice is that each chromosome has two sister chromatids. So each one is a copy of the same genetic material, the same DNA instructions joined together in the center. Okay, so I think you can just about see in this electron micrograph again that the chromosomes appear thick and two parts joined together. The other thing that happens during prophase is that the nucleolus disappears. Remember that the nucleolus is not actually a structure inside the nucleus of the cell. It's made up of loops of DNA and a lot of protein carrying out that very intensive work of making ribosomes. But when the DNA is condensed into chromosomes, there are no longer any loops of DNA available, so the nucleolus just disappears. In my example I used of thinking about a market where somebody is selling something at a good price and lots of people just go there, so that part of the market becomes very busy. Well, when everything is sold and there's nothing left to sell, people go away again, don't they? So you no longer see that condensed group of people. The centrosomes move to opposite sides of the cell. So remember that centrosomes are the organizing centers of the spindle, and the spindle fibers start to grow, to extend from the centrosomes. We'll have a look to see how that happens later. Okay, so here are the two spindle poles, the thickest part of the spindle, if you like. Okay, so prophase, the cell is getting ready to divide, but it isn't actually yet moving the chromosomes anywhere. Okay, the next stage is prometaphase, and this is the stage where the nuclear membrane disappears. So the chromosomes are no longer inside the nucleus, they're no longer separated from the cytoplasm of the cell. So remember that the nuclear membrane is made of a lipid bilayer with lots of proteins in it, especially the nuclear pores. What tends to happen is that the nuclear membrane forms little spheres. So we'll talk about how membranes can reassemble in a few lectures' time. But the nuclear membrane is no longer surrounding the DNA, but all the lipids are still present as little spheres inside the cell that can very quickly reform and remake the nuclear membrane after cell division. So if we think about what the chromosomes are doing, in prometaphase, the proteins attach to the kinetochores. So the centromeres of the chromosomes are the places in the center where the two sister chromatids are joined together. The proteins attach to those centromeres to form a structure called a kinetochore, and the kinetochore allows the spindle fiber to attach to the chromosome. Okay, so now each chromosome is attached to a microtubule of the spindle at its centromere by way of proteins which make kinetochores. Okay, so if we look at our electron micrograph here, we can see that the chromosomes have started to become separated towards the two spindle poles. They're moving towards the two different ends of the cell. Okay, so the next stage is metaphase. Meta means middle, and it's a good way to remember that this is the phase where the chromosomes can be seen to be lined up in the middle of the cell, in the middle of the spindle structure. So the chromosomes are lined up in the middle with one of each of the sister chromatids pointing towards each of the sides of the cell, each of the spindle poles. So here is our diagram, and here is our actual electron micrograph. So here you can see the spindle poles, the microtubules of the spindle, and you can see that the chromosomes are lined up in a flat line in the middle. So this structure is sometimes called the metaphase plate, meaning just a flat object. Okay, I apologize for another interruption. So I think I was talking about the metaphase plate, which is the name given to the line of chromosomes down the middle of the cell during metaphase. So when the sister chromatids are pulled apart, this lining up makes sure that one of the sister chromatids, one of the copies of the same information of DNA, moves towards each of the spindle poles. Okay, so it won't be that these two will both move this way and these two will both move this way. It will be this one will go in this direction, this one will go in this direction, this one will go in this direction, this one will go in this direction. So each new nucleus will receive one and only one copy of each of the chromosomes. Okay, next stage is anaphase, which is where we see the actual movement of the chromosomes towards the spindle poles. So to move in opposite directions, the two sister chromatids have to become separated where they were joined at their centromere. Okay, so now each different sister chromatid has a centromere attached to a kinetochore attached to a spindle, pulling it in opposite directions towards the two opposite sides of the cell. So here is our animation diagram, and here is our actual electron micrograph. So you can see that the chromosomes are being pulled towards the two spindle poles. And we'll talk in a little while about exactly how this movement can happen. It's a process of pulling and pushing against the microtubules of the spindle. Next phase is telophase, and this is where the two sets of chromatids, sister chromatids, have arrived at the opposite poles. So here we can see one set and another, one set and another, and the spindle starting to sort of separate, and we can see that the spindle fibers are no longer sort of looking continuous across the middle. They're being pulled towards each of these spindle poles. So at this stage, the two sets of chromosomes become new nuclei. The nuclear membrane forms or reforms around each of the daughter nuclei. And because the membrane was already there in the cell, complete with all the nuclear pores and other proteins that are part of that membrane, this can happen very quickly. The little vesicles of membrane can just join together and open out to become new membrane. The condensed form of the chromosomes also breaks down so that the chromosomes become dispersed again. They return to being the more open form of chromatin, so that they're no longer so visible as condensed structures like this under the light microscope. The spindle fibers, the microtubules here, break down and disperse. They return to being monomer forms of the protein. We'll talk about that later. So telophase is basically putting everything back the way it was before, except for having two nuclei, each with its new set of duplicated chromosomes in it. And then the last stage, cytokinesis, is to divide the two sets of chromosomes into two new cells. So the rest of the cell contents is also divided up. All the cytoplasm and the organelles inside the cell is divided in between the two new nuclei. So here is our diagram from the video, and here is an electron micrograph. So you can see the chromosomes, the DNA inside a nucleus here. You can see the spindle fibers are now becoming the cytoskeleton, going out from a nucleus towards the cell membrane, rather than the spindle fiber, just the last part of the spindle here, divided between the two cells. And the cells are pinched apart by an actin ring, another structural protein which is able to move and contract. It sort of pulls in and pinches those two cells apart to make two separate cells which can separate. In plant cells, because plant cells have a cell wall, a new part of the cell wall has to be made in between the two new cells, and that's called a cell plate. Okay, so that is the stages of mitosis and cytokinesis. I've just got a little video now that I hope will help you to remember and to understand these things, and I hope that you will enjoy it as we take a little bit of a relaxation time.
Every cell goes through it, the cell cycle, growing phase, synthesis, and then more growing. In mitosis we divide through cleavage, and we know that with the song we can teach it. The cell cycle, the cellular cycle, and we know that with this song we can teach it. G1S phase, G2M. And we know that with this song we can teach it. Give me that chromatin, give me that growth factor. Proteins love my style, at my goal to get wild. G1 grows those cells, organelles duplicate, and CDK phosphorylation helps regulate. S phase, replicate DNA, helicase, the unzipping those GEs, DNA polymerase, the adding nucleotides. If DNA is damaged, cycle twist to the side. The cell goes through it, the cell cycle. Growing phase, synthesis, and then more growing. In mitosis, we divide through cleavage. And we know that with the song we can teach it. The cell cycle, the cellular cycle. And we know that with the song we can teach it. G1F phase, G2M. And we know that with the song we can teach it. Keeping on growing. I'ma make it going. Mitotic cycling, dephosphorylase, decay. This is how we check if everything's alright. Take that cell to end phase, baby, let's see you divide. End phase, divide it up. When MPX around cells, baby, starting up mitosis. In the checkpoints, make sure that everything's attached. With proper segregation, we get two cells that match. The cell goes through it, the cell cycle. Growing phase, synthesis, and then more growing. In mitosis, we divide through cleavage. And we know that with the song we can teach it. The cell cycle, the cellular cycle. And we know that with the song we can teach it. G1S phase, G2M. And we know that with the song we can teach it. We1 kinase, make you put your phosphate on. Make you put your phosphate on. Tyrosine 15. CAK, make you put your phosphate on. Make you put your phosphate on. A1, phosphate. Take that phosphate, take it off. Take that phosphate, take it off. Tyrosine 15. Hell yeah. Activate, separate competition in your aspect. Every cell goes through it. The cell cycle. Growing phase, synthesis, and then more growing. In mitosis, we divide through cleavage. And we know that with this song, we can teach it. The cell cycle, the cellular cycle. And we know that with this song, you have learned it. G1S phase, G2M. And we know that on the test, you will ace it.
Okay, I hope you enjoyed that song, and I hope that it does help you to remember. It's the reason that I always have to say G1S phase, G2M, like that. Okay, so I always hope that some of my students will produce something similar. If you're a person who likes writing songs, singing songs, it would be a great way of helping you to learn this material. Okay, so the last thing I want to cover in this lecture is to talk about the details of the spindle. I've been talking a lot about what the spindle does, but how does it do it? Well, the spindle is a structure made of microtubules. So how are microtubules able to move things around in the cell? We've talked about the spindle pulling the chromosomes towards the two poles of the cell. How are they able to do that? How are they able to push the poles apart? The spindle sort of lengthens the cell, makes it longer and thinner. And how are they able to pull the chromosomes towards the poles? So the spindle is made of microtubules, and microtubules are made of a protein called tubulin. So I hope that you've already seen or attended the lecture about protein structure. So you know what I'm talking about when I talk about quaternary structure and so on. If you don't, please make sure that you do watch that lecture and know about it. So tubulin is a molecule that has a quaternary structure of two different subunits called alpha tubulin and beta tubulin. Very similar structures, but not quite the same. Okay, I'm noting a difference in the helix here, for instance. So tubulin is a heterodimer, which means it's a dimer, it's got two subunits, and they're different subunits, hetero. Okay, so tubulin is a heterodimer. It has a quaternary structure of two subunits associated together, interacting with each other at this interface in between them. So these tubulin molecules can come together to make long filaments. So this is rather similar to what we saw in keratin, where the molecules assemble end to end and side to side. Tubulin is another structural protein, another fibrous protein. So in the filaments, the tubulin molecules always arrange so a beta tubulin is making contact with the next alpha tubulin. Okay, so they're always in these alternating arrangements, alpha, beta, alpha, beta, alpha, beta. We never see one flipped around. We never have alpha, beta, beta, alpha. So this means that the two ends of the filament are different. There is one end which ends in an alpha tubulin, which is called the minus end, and another end that ends in a beta tubulin, which is called the plus end. Okay, so these filaments or protofilaments of tubulin can assemble together side by side, again similar to keratin, to form a cylindrical structure. Okay, so that's what we're seeing here. So you can see that they're sort of packed together to give a sort of spiral shape around a hollow core. So here's an electron micrograph taken from the end, and here's a diagram showing that the filaments are arranged in a circle with a space or a lumen in the middle. And here is an electron micrograph taken from the side, just showing the long chains of the protofilaments lined up. And this hollow tube structure is a strong structure. It's a very strong structure. It's stronger than it would be if the same proteins were arranged without a hole in the middle. Okay, if you think of things that are constructed, for instance, bicycles, they are often made from metal tubes with a hole in the middle, because that's actually stronger than it would be if the same metal was used in without a hole in the middle. Okay, so microtubules are a cylindrical structure. Okay, so how do microtubules produce movement? Well, I mentioned that they have a plus end and a minus end. And when the microtubules form part of the protein, the minus end is at the spindle pole, and the plus end is attached to the chromosome. Okay, so more tubulin is added to the plus end than to the minus end, and more tubulin leaves the minus end than the plus end. And it's a dynamic process, so it depends on the balance of tubulin molecules being added to the microtubule and tubulin molecules leaving the microtubule. But when the spindle is actually pulling the chromosomes, more is leaving than being added. So there's a movement in this direction. And when the microtubules are growing towards the chromosome, more is being added than leaves. So there's a movement towards the chromosomes in this direction. There's a note here talking about speckles. This is an experiment which shows this movement. Some of the tubulin molecules are labeled with a fluorescent label, and when that's looked at, it appears that those speckles are moving as the microtubule grows. So here's the speckles. Here is where addition and removal of tubulin molecules is happening at the end. So if more is added here than leaves, the speckles will seem to move in this direction. If more falls off than is added, the speckles will seem to move in this direction. Okay, so altogether we have a microtubule fiber, a long fiber, but the actual tubulin molecules which make it up keep changing. They keep being added to one end and removed from the other end. So when we look at how that works in the actual spindle setup, here are the two centrosomes which act as an organizing and starting point for the spindle. So this is what we can see in the microscope as the spindle pole, the place where all the spindle fibers seem to be joining together. There are different sorts of microtubules doing slightly different jobs. So the astral microtubules are going out towards the cell membrane of what will be the two new cells and starting to be the beginning of the cytoskeleton. The kinetochore microtubules are the ones which actually attach to the chromosomes and are able to move them. So here is our metaphase plate of chromosomes. Here are the kinetochore microtubules attached, being shown in blue here. And in red, there are interpolar microtubules, and these are illustrated as with these black dots, which represent a motor protein. So this is a protein which can produce movement. So if we think about the spindle poles, if these microtubules move so they're slid away from each other, and because the microtubules are stiff and strong, they will push the two spindle poles apart, and they will lengthen the cell, make it longer and thinner, ready for cell division. So if you think of my two elbows here being the spindle poles and my arms being the microtubules, I'm pushing them apart. Okay, and these motor proteins, if you think of them a little bit like a motor going round and pushing these fibers apart. Okay, so lengthening of the cell because of the interpolar microtubules. The spindle fibers themselves are able to pull the chromosomes because here at the minus end, at this stage, there will be tubulin leaving, and there's an attachment to the kinetochore at this end, so less tubulin will be being added to this end. So the tube will be getting shorter and shorter, and that pulls each of the sister chromatids towards one of the poles. Okay, so there's a pushing movement, lengthening the cell, and a pulling movement, moving the chromosomes. Okay, so do ask any questions on that. That is all I've got for you today. I'm sorry if there's been some interruptions in this lecture, but I hope that you've been able to follow well. if you want to know more, I think that this book is the best one to go to. It's the one I've taken a lot of the diagrams I've used from. You should be able to find it at this link. And chapter 17 is about the cell cycle. And for next time, we'll be looking at apoptosis, which is covered in chapter 18. So please do get that book and read and check what I've said and make sure that you understand what I've been talking about. Okay, so I will see you next time, or at least you will see me again, when we will continue to think about cell division and cell death.